Okay, this is talking about a gram stain. Gram stain is a differential type of stain. It is the most important stain that's used in microbiology to help to distinguish between your gram-positive bacteria and your gram-negative bacteria. How is it able to do this? It has to do with the structural differences in the composition of the cell wall. Specifically, it's going to look at how thick is the peptidoglycan layer that's in the cell wall and how it reacts to some of the different chemicals of the gram stain. So a differential stain, remember, uses two different stains. In the gram stain, we're going to be using crystal violet as well as saffron red. When you have completed this and are viewing the slide under the microscope, you can determine the cell size, the cell arrangement, and cell morphology. So that first stain you use, which is known as the primary stain, is crystal violet. The second stain, that's saffron red, is the secondary stain, or you could also call it the counter stain. So in this slide, what we're showing is the gram-positive versus the gram-negative bacterium. And this darker brown color is representative of the peptidoglycan that makes up the cell wall. So as you can see in the gram-positive, it is a very thick layer. In a gram-negative, it is a very thin layer of peptidoglycan, and on the outside of it, around that peptidoglycan layer, there is a lipopolysaccharide membrane. This membrane is not present in the gram-positive bacteria. So this is going to play a huge role as to the way it responds to the stains. So to do the actual gram stain, you would have made your smear, you would have done the heat fixation step. Then what you will do is you will add crystal violet for 1 minute. As we said just a bit ago, this is your primary stain. After 1 minute, you will rinse that crystal violet off with water. Then you add the grams iodine for 1 minute. After 1 minute, you rinse it off. Then you will add ethyl alcohol, leave it on for 30 seconds, and then rinse that off. Now with the ethyl alcohol, this is a decolorization step. Some people, instead of doing for 30 seconds, they will do two 15-second steps. So they'll add ethyl alcohol for 15 seconds, rinse it off, add ethyl alcohol a second time for 15 seconds, and then rinse it off. And some people will just add the alcohol and just add enough until you see color running off and then stop. Whichever method you use, when you're done with the alcohol, you rinse, and then you add the saffron red for 1 minute. This is your counter stain or your secondary stain. After a minute, you're going to rinse it off and then blot it dry and then view it under the microscope. Now in this slide, it is showing once again these same 4 steps, but it's showing you if you were to view under the microscope after each step, what would you see? So it's showing here, first you add that crystal violet. All the cells would look purple if you viewed it right now. After you add the crystal violet, you add the grams iodine. At this point, all cells would still look purple. And then when you add the alcohol, what this is doing is a decolorization step, and you'll notice that some of the cells will remain purple, but others become colorless. It would actually be your gram negatives that lose the purple color. The gram positives remain purple in this 3rd step. And then your final step is when you add the saffron red, and now you notice that your gram positives are still purple, but the gram-negative bacteria, which in this particular schematic drawing are shown as the rods or bacillus-shaped organisms, they pick up that secondary stain of saffron red, so they're going to look pink. So as I mentioned, when you add if we go step by step, and what's happening is you add the primary stain, that crystal violet, which is purple, all the cells are going to look purple. The grams iodine, it is known as a mordant. This just means it helps to act as a stabilizer, and it is going to form a complex with that peptidoglycan. Excuse me. The crystal violet iodine complex basically forms a crystal in the cell wall. What's going to happen now is that when you add the alcohol, the gram-positive bacteria, that has multiple layers of peptidoglycan, the alcohol dehydrates the peptidoglycan, and so your crystal violet iodine crystals, they can't leave. It's like they're stuck in there. They can't get out. The gram-negative has a much thinner layer of peptidoglycan, but remember it also has that lipopolysaccharide membrane on the outside of the peptidoglycan. Alcohol will dissolve that membrane. The lipopolysaccharide membrane gets dissolved. It kind of ends up leaving holes in the peptidoglycan because the peptidoglycan is really only about 1 layer thick. And so there's nothing then to hold the crystal violet iodine crystal in, and it all washes out. That's why it loses the purple color. So that alcohol dissolves lipids that are in the membrane. The gram-positive bacteria do not have this outer membrane, so there's nothing to dissolve there. Instead, it dehydrates the peptidoglycan layer, making it tighter and holding those crystals in. So that's why your gram-positive, those purple crystals, the crystal violet iodine, those purple crystals are kept in, so it's going to look purple. But because you basically dissolved and ripped off that lipopolysaccharide membrane with the alcohol, and then washed out the crystal violet iodine purple crystal complex, the gram-negatives are going to look colorless. Now you add the counterstain of saffron red, which is a red dye. That saffron, the gram-positives already, all the spaces are taken with the crystal violet iodine complex, so there's no place for the saffron to go. But the gram-negatives are going to stain red or purple because you just washed in the previous step all the crystal violet out. So there's all this empty space there for the red dye to go, and so that's why the gram-negatives will stain red. This is just a different way of showing and comparing the two. You heat fix it, you add the crystal violet, you add the iodine, everything's still looking purple. You add the alcohol for the decolorization step. Your gram-positives are still purple, but now your gram-negatives are colorless. And then you add the counterstain. Gram-positives are still purple, but your gram-negatives are pink. And these are pictures of actual slides of your gram-positive, which are this darker color, and the gram-negatives, which are the lighter red color.